In this video, we are going to be talking about enzyme inhibition. But what actually is an enzyme? An enzyme is a specialized protein with extraordinary catalytic power. It allows them to speed up chemical reactions by lowering the activation energy, which is the amount of energy needed for a chemical reaction to occur, which would otherwise be too slow to keep us alive. Each enzyme has a special pocket called the active site, where its target molecule, the substrate, binds perfectly. Now think of this active site as a reserved parking spot, and the substrate is the car with the specific parking pass. When the car parks in the right spot, the enzyme is able to do its job, allowing the reaction to proceed. Enzymes are like helpful workers in your body. They have many different functions, but just like how workers can get stopped or distracted, enzymes can too. Today we'll explore two different kinds of enzyme roadblocks, competitive and non-competitive inhibition, and how these change the way enzymes work. Now imagine another car, one that looks really similar to the real car, speeding in and stealing the parking spot. The real car, which is the substrate, is left circling around with nowhere else to park. That's competitive inhibition. A competitor molecule that resembles the substrate binds to the active site instead, and the reaction slows down because the real substrate can't get in. But this can be overcome. If there's enough of the real cars, eventually one will grab the spot. That's why competitive inhibition is reversible, just by adding more of the substrate. A real-life example of this is methanol poisoning. Methanol becomes deadly when enzymes in the liver break it down, so doctors treat methanol poisoning by giving people ethanol. The ethanol competes with the methanol for the enzyme's active site, and since it binds better, it blocks methanol from being broken down into toxic products, giving the body enough time to remove it safely. But sometimes the problem isn't the parking spot being stolen, it's the parking lot itself being damaged. Now imagine a construction crew comes and blocks off part of the parking spot, changing its shape. Even if the right car with the right parking pass shows up, it no longer fits. This is non-competitive inhibition, where the inhibitor binds at a different site, usually known as the allosteric site, not the active site. And it changes the shape of the active site so that the substrate can no longer bind to the active site. Unlike competitive inhibition, adding more substrate cannot fix the problem because the spot itself has been changed. A real-life example of non-competitive inhibition is cyanide. It binds to the allosteric site of enzymes, not the active site that is used in the mitochondrial electron transport chain in cellular respiration. This changes the shape of the active site, inhibiting the process of the electron transport chain and stopping energy production. Even if oxygen molecules are present, the enzyme won't work. Non-competitive inhibition with cyanide can be lethal if not treated immediately. Why is enzyme inhibition even important? Enzyme inhibition plays a huge role in everyday biology and medicine. Inhibitors can be harmful, like poisons and toxins, but they can also be helpful, like many prescription drugs or blocking other toxins from binding. Actually, some of the most common medications, like aspirin and antibiotics, work by blocking enzymes. Enzyme inhibition is very important because it slows down or stops certain reactions, which can help us treat illnesses, relieve pain, and also fight infections. In conclusion, competitive inhibition is like another car stealing the spot. The enzyme gets blocked, but can be reversed if enough substrate are present to win it back. And non-competitive inhibition is like the parking spot being damaged. It doesn't matter how many cars arrive, none can park. Enzymes are a large part of life's chemistry, and the inhibitors remind us just how fragile and precise these processes within our bodies are.